Hello guys, welcome to our channel. Today's topic of my video is GPCR or G protein coupled receptor mediated inositol phospholipid signaling pathway. To know about the structure of GPCR and its proteins, how stimulatory G protein or GS activate adenylyl cyclase and produce cyclic AMP leading to the downstream signaling, then please watch my previous video, that is chapter 1, GPCR regulated production of cyclic AMP and PKA activation. Link is provided in the description. So let's start. Today I will discuss about GPCR or G protein coupled receptor mediated activation of a G protein called GQ, which activates inositol phospholipid signaling pathway. Many GPCRs activate this type of G protein that activates a plasma membrane bound enzyme phosphatidyl inositol specific phospholipase C-beta called PLC-beta or PLC-beta in short. This is the crystal structure of the enzyme. Activation of this enzyme mediates different responses in different type of cells. Here is a list of responses mediated by the activation of PLC-beta. The GQ protein activates the PLC-beta just like how GS protein or stimulatory G protein activates adenylyl cyclase, which is through the alpha subunit. Here we can see in the animation the activated G-alpha dissociates from the beta-gamma complex and goes to and activates PLC-beta. GPCR mediated activation of adenylyl cyclase and production of cyclic AMP has been discussed in my previous video. Before going into the inositol phospholipid signaling in detail, let's first understand how PIP2 is formed. At first, phosphorylation of phosphatidyl inositol or PI by phosphatidyl inositol kinase or PI kinase at the 4th carbon position results in the formation of phosphatidyl inositol 4-phosphate or PI4P. Then, PI kinase again phosphorylates PI4P at the 5th position to form phosphatidyl inositol 4,5-bisphosphate or PI4,5P2. Breakdown of this PI4,5P2 results in the synthesis of inositol 1,4,5-triphosphate or IP3 and diacylglycerol or DAG, which I will discuss in detail in the next animation. Here are the structures of IP3 and DAG. This PI4,5P2 is the least abundant phospholipid, that is about 10%, and also the lipid, that is about 1% in the plasma membrane. Now let's understand inositol phospholipid signaling pathway in detail. Here we can see a 7-pass transmembrane receptor G protein coupled receptor or GPCR. Binding of the signal molecule results in the activation of the receptor. The activated receptor now works as a guanine nucleotide exchange factor or GEF, which in turn activates the G-alpha subunit of the GQ protein. This occurs by removal of the GTP and binding of the GTP. This mechanism has been explained in detail in my previous video. The activated G-alpha now dissociates from the beta-gamma complex and goes and interacts with the PLC-beta or phosphatidyl inositol specific phospholipase C-beta. The G-alpha activates the PLC-beta, which in turn hydrolyzes the phosphatidyl inositol 4,5-bisphosphate or PIP2 into two molecules, that is inositol 1,4,5-triphosphate or IP3 and diacylglycerol or DAG. Both of these IP3 and DAG are second messengers, just like cyclic AMP mentioned in my previous video. Second messengers are intracellular signal molecules that are released by the cell in response to an extracellular signal molecule. This extracellular signal molecule is the first messenger. Inositol 1,4,5-triphosphate or IP3 is a water-soluble molecule that acts as a small intracellular mediator. It leaves the plasma membrane and diffuses rapidly through the cytosol. IP3 then binds to and opens an IP3-gated calcium release channels, also called IP3 receptors, present in the endoplasmic reticulum membrane. Opening of this channel releases the calcium from the lumen and thus raises the concentration of the calcium in the cytosol. Another hydrolysis product of PIP2 is diacylglycerol, which can perform different functions. It is also a small intracellular mediator which remains embedded in the plasma membrane, where it has several potential signaling roles. Diacylglycerol can activate a crucial serine-threonine protein kinase called protein kinase C or PKC with the help of calcium. Rise in cytosolic calcium due to the opening of IP3 gated calcium channels allows translocation of the protein kinase C to the plasma membrane from the cytosol, where it is activated by the presence of DAG, calcium and the negatively charged phosphatidyl serine present at the inner half of the plasma membrane. The activated PKC now can phosphorylate different target proteins. Only the conventional PKCs are activated by calcium and DAG, while the atypical PKCs are not activated in this way. In addition to the role of activation of PKC, DAG can also be cleaved to release a molecule called arachidonic acid. This arachidonic acid can either function as a signal molecule on its own or can be used to synthesize other small lipid signal molecules called eicosanoids such as prostaglandins. These prostaglandins participate in pain and inflammatory responses. So from this video we can understand the mechanism behind inositol phospholipid signaling pathway, how GPCR mediated activation of alpha subunit of GQ protein leads to the cleavage of PI45P2 into IP3 and DAG, how IP3 causes the release of calcium from the ER lumen, and how these calcium regulate the activation of PKC in the presence of DAG. To know about the structure of GPCR and its proteins, how stimulatory G protein or GS activate adenylyl cyclase and produce cyclic AMP and leads to the downstream signaling, then please watch my previous video, that is chapter 1, GPCR regulated production of cyclic AMP and PKA activation. Link of this video is provided in the description. In the next video, I will discuss about receptor tyrosine kinase or RTK. For more videos, please subscribe to my channel and don't forget to like and share my videos. Thank you. Stay home, stay safe and study online.